some of our students registered for the course but didn't like to sit here. It's okay.
It's okay. कोई बात, light is okay. और लाइट क्या जरूरी है? अच्छा, okay. Good morning. So okay. So for the first lecture, I'm going to talk very briefly about brain imaging and fMRI. So as I said, if you have any questions, you can just raise your hand and ask me. Okay. So I will start with a quiz, right? So all of you have some idea of what brain imaging is. How many of you know? Can you give me one example of brain imaging methods? Okay, you can sit and answer. Okay, good. MRI is one. Okay, diffusion is good. Yes, very good. Anyone else? Okay, what? Okay, so that's like MR related, right? Anyone else? Good. Okay. MRI, CT, F nears. What? PET. Okay. Anyone else? Very common. X-ray. Very good. Okay. Um, anyone else? What? EEG. Okay. Okay. Yeah. EEG is good. Anything? Thermal imaging. Thermal imaging. Okay. That's. Are we missing something very simple? Hmm? DTI is MR related. Like every person, I guess most of you had MRI, or maybe your mom had ultrasound. Have you heard? Okay, good. All right. So the first is a quiz. So these are four imaging modalities, right? So A, B, C, D, right? How many of you can guess which modality is what is A, what is B, what is C, what is D? Okay. A is what? A is structural MRI. No. Um, okay, if you did you say C is MRI? Good. Okay, C is MRI. Um, anyone? What is A? What is B? Hmm? What did you say? Say. No, no. Um, any? Okay. Okay. I have already like we have already covered every single modality. So what is A? Okay, I will give you. Okay, make a guess. I can answer one of them which you want. Okay, anyone? So those of you who have like registered for the course, doesn't matter, who want to participate in that data analysis, like please email me at this address and just like say A is CT, B is this, C is MRI, and what is D, okay? So you have maybe 1 minute, just send me. I need your Gmail addresses because I can share like a Google Colab link to do the analysis, okay? Can anyone guess? Okay, I'll do this. If anyone can guess, I will buy all of you ice cream, okay? Make you would become so popular with your friends, right? Anyone? Very good. So A is CT, B is PET scan, C is MRI. What is D? You have a minute left. All right. Okay. Time's up. Okay. Unless you want more time. Okay. So I don't think any of you could have guessed. So A is CT, B is PET, C is MRI, and D is actually a person's digital photo of, like as someone said, histology, right? So again, this is a picture which was taken around 30 years ago, and it was called the Visible Human Project. A person um, who was on the death row for having committed some crime. So before he was executed, he decided to donate his body to science. And these were the state of the art imaging methods then. So CAT scan was done, PET was done, MRI was done. At that time, there was no fMRI or DTI was not that popular. So after this, then after the person was executed by drugs, so then like his whole body was cut into thin slices, and then digital picture was taken. And I still find it so amazing that even with technology that was 30 years old, like especially the MRI and the actual photo was so similar, right? In some way, I would say the MRI looked even better than the actual photo. I would also say that now the CT scan or even the PET scan, they look much more like the digital photo. And with MRI, now you can get even better spatial resolution and temporal resolution, right? So so these are so CAT scan and MRI are what is called structural images, right? They are like looking at like we are acquiring a 3D data set, while with PET scan, it's more like a functional or a metabolic change. And one of the question always is, can we ever understand the structure and function and how they are related, right? So this one, all of you know what this is. All of you, there's already a hint, right? So what is this one? Hmm? I have, there is honey written there, right? So what is this thing? Hmm? Say that, the what of honey? It's not extraction, it is the beehives which are grown, right? And so they are placed, but what is this drum doing? Very good, right? So you use centrifugal forces to extract the honey, right? Otherwise, it's not that easy to extract it, right? So then the question is, if you could get a very good image, right? If you could look at a structural image, do you have enough information to find the functional information or the underlying function, right? And so in this case, um, if instead of a cylinder, if it was a square, would you have said it would use centrifugal forces, right? Um, so that is like, I feel like one of the fundamental challenges of brain imaging, that is, we can get very good description of the structural information, but we have to find good functional information, right? And sometimes we can extract some information knowing the structure, but sometimes that is never enough, right? Um, so all of you have a smartphone, I'm guessing, right? So how much megapixel does your cell phone has? Hmm? 12. Oh, okay. Um, so 12 megapixel, if you made a matrix, what does that mean? Is, is that the definition, or the definition is if you made an image, how many voxels would that image use up, right? Maximum would be 12 megapixel, right? So so similar, again, I'm going to talk more about it as we cover. So maybe um, one explanation is you can draw, you know, use MRI and so on to get a picture of anatomy, or else you could also get information where you can find what does different brain area look like, right? And this is like one of the first study about brain imaging experiment. This is by an Italian physiologist by the name of Angelo Mosso. And what he had proposed was that if you want to like study function, what you could do is you could make someone lie on a seesaw, and during baseline condition, they would be like, it would be balanced. And when they did some complex cognitive task, then there would be more blood flow to the brain and it would tilt. So that is how you would know this person is doing some complex task, right? And although you may think it is so like naive and so on, that is the basic principle for fMRI, right? So again, as I said, with MRI, you take several minutes to acquire an image, right? So you take several minutes to collect, let's say, 500 by 512 matrix with 1 cubic millimeter spatial resolution, and then maybe you could collect 50 or 70 slices. But with fMRI, what is done is you collect the same information at a much faster rate, right? So this one is taking several, okay. Is there a, okay. So the one on like the MRI image would take several minutes to collect. About, I think it's okay. I think it's okay. So the MRI would take several minutes to collect, while with fMRI, what we can do is, oh, thank you. Yeah, so this is MRI, this is fMRI. fMRI would take, instead of 1 cubic millimeter, this is 3 by 3 by 3, and the whole volume could be collected every 2 seconds, right? But now you can collect it even faster. And the basic idea is with fMRI is dependent on bold oxygen level dependence, called the bold signal, as someone had mentioned. And the bold signal, the idea is when you perform some neuronal activity, there would be increased firing at specific brain regions, right? If you are tapping your finger, there will be increased firing in the sensory motor cortex. When there is increased firing, it will use up the oxygenation, and so there will be vasodilation, which will lead to increased blood flow, right? And when there is increased blood flow, there is more oxygenated red blood cells that come in compared to deoxygenated red blood cells. Oxygenated red blood cells and deoxygenated red blood cells have different magnetic properties, and therefore with fMRI, you can see an increase in signal. Right? And this is the first fMRI study that was done by Ken Kwong. They showed a visual stimulus, like a flashing visual stimulus. So this is baseline condition, right? So the first 60 seconds is off period. Then the next 60 seconds is the on period, right? And then again it alternates. And this is how the signal from the visual cortex, which is around here, looks like, right? Baseline, on condition, again baseline, and on condition, right? So this is the off condition, there is no change. During on condition, you can see increased intensity here. Again, during off condition, there is, again, back to baseline condition, again during on condition, the signal goes up, right? So when you perform different tasks, then in specific brain regions which are involved with the task, there is an increase in signal intensity. So some of you are studying electrical engineering, or some of you, Dr. Bhumika had mentioned that some of you are from IIT Madras. So it doesn't matter from where, but can anyone explain what is a linear time invariant system, LTI? Uh, no one has read about, anyone has taken signal processing, signals and systems? No? Okay. Okay, so okay. So the idea is that when you perform a task, so what it means is that if you have an input, you present the input and there is an output, right? And what a linear time invariant system means is that the input output relationship is always conserved, right? So if I tap right now and there is a change, then if I did the same thing 1 hour later, the response would still be the same, right? If I did it tomorrow, it would still be the same, right? So that is time invariant. Or if I did, you asked me to be on off state for 120 seconds, and then you did the on state, then the signal also will be off state, no changes will occur till I tap my finger. Like so, the first 120 seconds will be baseline, and only then the signal will change, right? So, so that is time invariant. The linearity means, like in finger tapping, the analogy would be if I tapped like this and the signal changed by amount X, and if I tap twice, the signal changed 2 times, then it's a linear system, right? So while it may not be always valid for most of the data analysis, we can use this as an assumption. Okay, so this is the basic idea with functional imaging, right? You like that these are linear time invariant system. So if you scan someone, and then if you scan them the next month or the next day, the response should be similar. Okay. So this is a picture of how do we compare the different imaging modalities, right? So this is in seconds, the temporal resolution. This is the spatial resolution, right? So for example, patch clamp or light microscopy or single cell unit recording have very high spatial and temporal resolution, but they are highly invasive, right? You would have to drill a hole in someone's brain to study. MEG or EEG, while they are non-invasive, their spatial resolution is not very good, right? The like PET imaging is like the spatial resolution is not very good, like in tenths of seconds. Their spatial resolution is also lower compared to fMRI, right? So fMRI is around here, so it has higher spatial resolution than PET imaging, and also has better temporal resolution than PET imaging, right? So That is what makes fMRI unique to study brain function. So before, the way to study brain function was to um, cut open someone's skull. So obviously it wasn't done in healthy subject. It was typically done in people with epilepsy or tumor. And what would be done is that you would identify that, say that, okay, 1, 2, 3, 4 are the 4 regions of the sensory motor cortex. And then one would basically stimulate the specific region. And what would happen is, if you stimulated their hand region or tongue region, those the patient would move their hands, right? So these are like uh, uh, so these are called direct cortical stimulation, right? And then slowly there is more and more sophisticated method with an array where you can like stimulate very fine structure and see how the function changes, right? These are other ways in which um, we can do the stimulation and then overlay on the MRI region to identify the associated functional regions, right? Again, this is PET imaging, where in PET imaging, you give a tracer, right? So you inject a radioactive tracer, and the idea in PET imaging is that when you perform a task, because there is more blood flow, so by simple probability, more of the radio tracer will go to the region where there is more activity, right? So that is how one could identify. Also, with tracer, you would have two conditions like task 1 and task 2, right? Task 1 could be just baseline condition where they are not doing anything. Task 2 would be where they are doing finger tapping or visual stimulation, and then you would compare the two different conditions and then identify where there is increased activation. The other ones are like, as I mentioned, is like EEG, where you place the electrodes. Again, this is on the surface, and then you can get the EEG signal, and then you can do the Fourier transform to look at the different frequencies. Do all of you know what a Fourier transform is? Yeah. Can someone tell what is Fourier transform? Yes. Okay, but how does like what does I understand what you said, but how does Fourier transform go about doing that? Okay, very good. So what a Fourier transform does is it takes any signal and decomposes into a set of sine and cosine waves. What is your name? Sahil. Okay. Um, so it will take any signal into decompose into sine and cosine, and because once you can decompose a time series signal into a set of sine and cosine waves, then it is much easier to represent those, right? So just as an example, if you measured um, my like someone's heart rate, let's say you used a physiological measure, which is measuring someone's heart rate and respiration rate. So what you would get is like one sinusoidal signal representing the heart rate and another sinusoidal representing the respiration rate. The rest would mostly be noise, right? So now to represent a sinusoidal signal, how many parameters do you need? Sahil? To represent a sinusoidal signal, how many parameters do you need? Okay, what are those? Okay, there is, how about the phase? So, so there is three parameters. So amplitude, frequency, and phase, right? So suddenly a large time series is now decomposed into two components, and each one you need only three parameters to completely describe it, right? So our analysis becomes much easier, right? And that is also a way to clean our signal. Um, so we will be, again, I talk about Fourier transform because when we do fMRI, we will be talking about the frequency spectrum, which is in the fMRI signal, right? So these are the components. And again, one can use EEG and fMRI to localize where these components are um, in the frequency, but also localize them in space. Another imaging modality is called magnetoencephalography, called MEG. And in EEG, where you are looking at electric potential, here you are looking at magnetic potential, right? And it is very similar, and there are some added advantages compared to EEG, but it is MEG system is much more expensive, but we can identify dipole and active neurons, so you can define very finely tuned differences in these components. And this is how a signal looks like. And then you can use this to map out different components, right? And by doing this, for specific tasks, what you can do is you can make these components as to when there is a, if you're presenting a complex stimulus, right, where based on, let's say, I asked you to, I was showing you a movie and asked you only tap your fingers when, let's say, your favorite actor is present on the screen, right? So then by using this, you can look at the visual input, the motor input, the premotor input, and then analyze how they are connected to each other. Okay. And then look at like these changes, right? This is how fMRI setup looks like. The fMRI setup, like the fMRI also uses the MRI machine, right? There is only a few additional component, right? So you have a video screen for presenting the visual stimulus, headphones to get auditory. This is button press to perform specific task, right? So using this, you can present three different kinds of stimulus, and then you can collect the data, right? And this is where a technician or a researcher can design which slices of which regions of your brain to focus. And the MRI again has three main components: the main magnet, the gradient coil, and the RF coil, right? So these are the three main components of an MRI machine, which is used to like magnetize specific protons, localize them, send and receive signal, and reconstruct, right? So those are the main components. And the MRI actually stands for NMRI, right? So this is NMR stands for nuclear magnetic resonance, and it was first discovered by Bloch and Purcell, for which they won the Nobel Prize in 1952, right? And so nuclear is property of nuclei of atoms, M is magnetic field, and resonance is interaction between magnetic field and radio frequency. And people excluded N part because like there is this connotation of nuclear bomb, right? So the history of fMRI basically started with Seiji Ogawa, who discovered the Bold effect, right? And he suggested, or he observed that Bold effect with T2 star Bold vessels became more visible as blood oxygen decreased. Jack Bellevue observed the first fMRI using contrast agent, just like PET imaging, and then Ken Kwong published the first paper using Bold, right? And so this is what Seiji Ogawa showed, right? Seiji Ogawa showed that when there is oxygenated blood, there is no signal loss. However, when there is deoxygenated blood, there is signal loss, right? So now you have a better idea why Bold works, right? When there is task activation, there is, right, there is more oxygenated blood compared to the baseline, compared to rest condition, right? So there is, during baseline condition, there was signal loss, but during task activation in specific brain regions, there is more oxygenated red blood cells, so there is relatively less loss, so therefore we see an increase in signal intensity, okay? Like the good thing about, or the genius of Seiji Ogawa's paper was that although he had showed structural images, he had written in the discussion part that while he had shown this in structural images, if and when technology allowed us to do dynamic images, this could be used to study brain function, right? So many people think that it's just a matter of time before he also gets the Nobel Prize. So this is again showing during baseline condition, there is red blood cells, right? These are the red blood cells, and the purple ones are the deoxygenated red blood cells, right? And during task activation, there is more oxygenated red blood cells compared to deoxygenated red blood cells. So there is less signal loss, so therefore an increase in signal intensity. And while there are like many different models, how do we go from neuronal firing to Bold oxygenation? Most of the people, they agree that it is through when there is stimulation, there is more neuronal activity, which leads to excitatory activity and inhibitory, right? And then with neurovascular coupling, and then there is hemodynamic response. And these are blood flow changes, and then these are detected by MRI signal, and this gives rise to the signal, right? And that is how we can get. This also tells us how can we modify our signal, right? How can, if we need more signal, maybe we can feel, if we can go from 3 Tesla to 7 Tesla, there will be more signal change and so on, right? Can anyone tell what would happen? So let's say I was doing fMRI with 3 by 3 by 3 millimeter spatial resolution. What would happen if I made my spatial resolution 2 by 2 by 2? So my spatial resolution would increase. What would happen to my signal changes? Let's say, what would happen to signal to noise ratio? Would it go up? Would it go down? Signal to noise ratio would go.
Because size is small, so we are capturing different signals from the phone, like the
Right.
And in this case, because the space is less, we
Okay.
will also capture less noise compared to the first one. Signal to noise usually improve.
Okay. What is your name? Paul.
Paul.
Paul. Okay. Okay, but the signal to noise, the noise would go down, but your signal would also go down, right?
Yes. Yes.
So then what is the net effect?
So, sir, in terms of numbers, it is very much.
Okay. Okay. Okay.
I was talking in terms of
Okay. Okay. Good. Good. Yeah. Did you say increase? It would go down? Okay. It would. But anyone else? Yeah.
Also, because just a conductor, because of more space, there will be more averaging of signals. So for more signal, less space. For more refined signal, less spatial.
Okay. So do you think it will go up or go down? The signal to noise ratio.
With high resolution, it's not just
Okay. Anyone else? And anyone else? So the signal to noise ratio. Okay. I will tell you that the signal to noise ratio, when the spatial resolution increases, will go down, right? But can someone explain why? I wish there was a pen. It's okay. It's okay. There is one, or a highlighter. It's okay. So maybe so so what gives rise to signal? So what happens when we average, right? What happens when we average a signal? If we average a signal, the signal goes up, right? Why does it go up? So let's say um, I was collecting an image, and I collected the same image 4 times, and then assuming that the person did not move, I simply averaged them, right? I averaged 4 images. The signal to noise ratio will go up, right? Because my signal stays the same, but the noise as I add them up will go down, right? All of you agree with that? Sahil, do you agree?
Sir, would that be because lower the spatial resolution and that voxel will fluctuate? It will shape and origin.
Uh, mm, if you reduce the spatial resolution,
Spatial resolution that there are more chances that during taking the image, there will be a little bit fluctuation point because
Right,
the
right, right.
in the voxel and that increase the noise of the signal.
But I can, I actually, I would argue a little different, right? I would say that, let's say you had, let's say, uh, 3 by 3, and then suddenly we had the capability to do 0.3 by zero point 0.3. Then what do you think? Do you think it's still valid? Because when you have 3 by 3, and let's say someone moved by zero point 0.1 millimeter, right? So that's a tiny fraction. But if I'm doing 0.3 by zero point 0.3 and a person is moving by zero point 0.1, then there is a 30% mismatch from one image to the other, right? So, so it would affect more. Um, Let me see if I can try to explain. Right, so let's just assume let's assume a 2 by 2. So let's say there was signal here, but no signal here, right? So when we average, right, there would be signal, but what I'm doing is the noise from this voxel and this voxels would get cancelled out as an average, but the signal would stay the same. So typically, it would, the signal to noise ratio would go up. But I think what Paul Paulomi, okay, great. What Paulomi is saying is that what she's describing, she's not describing signal to noise ratio, she's describing contrast to noise ratio, the ability to differentiate between two states, right? So in that case, when you have higher um, spatial resolution, the contrast to noise ratio would also become better, but that is the trade-off, right? With higher spatial resolution, the signal to noise ratio goes down, but the contrast to noise ratio would go up, because in this case you could identify that there is one voxel that is not active, but 3 voxels which are active. Okay. Um, and again, this is a schematic of what we measure, right? So what we mostly measure is not here. What we measure is mostly here, right? So fMRI, as I had mentioned, is an indirect measure, right? And it doesn't look at, or it's not very sensitive to arteries, but more to venules and the veins, because the arteries are highly vascularized, and most of the time they are, um, like during COVID, right? When you did the test, how much did you expect your oxygenation to be? Very? No, then they would have sent you to the hospital, right? Very like above 90 generally, right? And that's because the signal that you get is from the arteries, right? And it's very well vascularized, and therefore it is mostly above, I would say, 97, 98. Um, on the other hand, the venules, right, they change a lot, right? And during rest versus activation, those regions change, and that is what we detect. So fMRI detects mostly from the venules, okay, and not from the arteries. And this is how the structure of the brain looks like, right? So, like this is first highly vascularized structure, and then I think I keep it's highly vascularized structure at very high spatial resolution, like here it is around 25 micron radius, right? So the other thing to mention is that what we measure with fMRI is not like, okay, this vessel is going this way, or this um, venue is going this direction. All we can say is, um, you know, a global trend. Okay. Um, okay, I'm not standing on anything. Okay. So this is maybe 1 millimeter by 1 millimeter, and yet this is how complex the structure is. So the other thing is that we are looking at global changes, right? So this is a study which was done, which shows changes in bold glucose use, oxygen use, and oxygen availability, right? And so basically the fMRI signal that we measure, which is very sensitive to blood flow, perhaps best describes the oxygen availability and the glucose use. Okay. And this is a very famous paper that was done by Nikos Logothetis, where in a monkey they implanted electrodes and did fMRI. And what they found was that these signal changes, the fMRI signal changes, was more correlated to local field potential, and therefore reflects postsynaptic potential similar to EEG and MEG measure. It was less correlated to multi-unit activity, which reflects action potential, which is similar to electrophysiology measure. And their point was that fMRI is more similar to EEG than to electrophysiological measure, right? And again, there was another study which was done using electrophysiology and fMRI, where a subject who had tumors, right? They, like, electrophysiology was done, right? This is hand representation using electrophysiology, and this is hand activation using fMRI, right? And this is face representation, and this is the face representation using fMRI, right? So there is like an overlap, but maybe fMRI is not very precise, um, but has like a overlap. Okay. So fMRI measures the population activity, right? So, and this is also dynamic in nature. So when you have, if you ask, like if you gave a task where you had to come up with a number of words, starting, let's say, with a letter S or something, right? Or any letter, and then ask the subject to quietly generate those letters, and then off condition and on condition. If you did a task for a few minutes, you would find activation like this. But if you ask someone for a long period of time, then there was no significant changes, suggesting that, you know, if I asked you to generate like verbs starting with a particular letter, you most likely will run out of words by a certain number of minutes, right? Very few people can still generate verbs after 15 or 10 minutes. So there would be no activity, right? So the bottom line is that in spite of all its limitation, right, fMRI is still the most popular method to study brain activation. Like bold imaging can resolve activation at a fairly small scale with high spatial resolution. Postsynaptic potential and action potential are correlated. So either way, what we are getting is somewhat meaningful, right? Even if bold activation doesn't correlate completely with electrophysiology, that doesn't mean it's wrong, right? So I think I will take like a 5 minutes break. If again, like if you have any question, I'll try to answer before I continue. I will also check my email. Those of you haven't, please send me your email addresses, or if you prefer, you can also write it in a piece of paper. Okay. I have 1, 2, 3, 4, 5, 6, 7. I have 13 students. If you are shy, you can write it in a. We can also like write it on a piece of paper, maybe. If you don't, you can block me after the course, no problem. Any questions? Anyone? Yeah. Of course. Yeah. So the question was, could it be because of habituation? Absolutely. So maybe habituation would have, I should have said habituation first, and then maybe after a certain time there would be. So maybe I described it poorly, but yeah, that's what I meant, like habituation.
What could be the habituation also, and also lack of effort? I mean, as you said,
Right. Yeah, you give up. Yeah. Yeah. What is your name?
Sir,
Rahul. Okay. Okay. Anyone else? Any other questions? All right. Okay. Again, if you have any questions, feel free to ask. No.
This image you talk about which PET MRI vaccine, and also do we go with PET fMRI something?
So that's a good question. So there is, so PET CT means that the machine has a PET and a CT in the same room, right? That is what it means. PET MRI means that the machine has MRI and PET in the same machine, right? But PET itself is a different technology, right?
In PET MRI can we do fMRI?
Yes, yeah. So there are a few groups which do PET MRI or PET fMRI. It's just that, does anyone know how much MRI cost, a 3 Tesla MRI?
8-9 crore.
Maybe Dr. Bhumika would know better. Right. Yeah. Right. Okay. And the PET machine is maybe 3 times more expensive. And so just combining them becomes even more challenging. So the PET, as I mentioned, does not have good spatial resolution. So therefore, because you're spending like 50 crores on a PET machine, like adding 1 or 2 crores to have a CT inserted also is not that expensive. So they do like PET CT, right? So they use the CT image for reconstruction of the PET data. And so that is what it is used. Did I answer your question?
Sir,
Yeah.
I have a very trivial question.
Yeah.
I think that what I can write down is the signals right the geospatial.
Yeah.
So, sir, does it really show that these areas are active, or does it show that these areas are just consuming more blood? It can also happen that some areas are more efficient that consuming blood.
That's a very good question. Um, so I think what you are asking is called causal relationship, right? And that is like a causal relationship would be like, let's say, um, all of your moms, right, would say, study hard, because if you study hard, your grades will go up, right? So that's like you're making a causal, okay, you're making a causal relationship that if you study harder, your grades would go up. Um, but but from um, another way one can think is that, okay, if you study harder, right, then you would be awake longer, right, and therefore you would drink more liquid, right? So that also has a good correlation, right? The more amount of liquid someone is consuming, maybe their grades would go up. But that is like, it has a good correlation maybe, but that is not a causal relationship, right? So in terms of fMRI, yeah, technically that is true, that this is not establishing that, you know, because you tap your fingers and there is activity in the sensory motor cortex, it doesn't prove that, there is a direct pathway. um, so that is true, but generally we assume, right, functional imaging assumes that that is the case, and therefore um, what we see, therefore we called it correlation, right? Tapping a finger correlates to voxels in the sensory motor cortex. If you see the visual flashing checkerboard, then there is a correlation with the visual cortex. So what we are seeing is more a correlation effect and not a causal effect. Although, if you did it in thousands of subjects, it would suggest that it plays a very important role. Okay, any other questions? Okay. So the next thing I want to cover briefly is the hemodynamic response function, right? As I said, the brain activity that we see is because of neuronal firing and the underlying hemodynamics, right? And this is how the signal looks like when you when you perform a task. There is something which shows the initial dip, right, when there is more oxygen consumption, and then the signal goes up, and as the stimulus is on, it will stay up, and then when you stop doing the task, the signal will go down and then again go back. So this is called the post undershoot, which people have some ideas, but I don't think anyone understands. So this is the idea, right? So you have neuronal firing with the hemodynamics, and that is how the signal changes, right? And so you can have like I showed you for finger tapping, you could have more complex tasks like objects, faces, and places, and then you can identify in the brain which are associated with objects, which are associated with faces, and which are associated with places, right? And in this case, the task design that you have to design has to be more complex. It cannot be just off and on, like it cannot be like off period and another stimulus, right? Here you have to do more complex subtraction, right? So here is like a simple setup, right? So let's say I want to design a task where I want to study what would happen, are there brain associated with objects? So if I did simply like blank screen versus intact object, and I did my analysis, I would find some region, but would that be associated with intact objects? Right? What could be the confounding errors? Anyone? Yeah. Oh, make a guess. Make a guess. What is your name? I can't hear you. Sanchita, okay. Okay, so I want to study, I want to design a task to study in which brain regions are associated with intact objects, right? And my question is, if I just showed a blank screen followed by intact object, then blank screen followed by intact object, and if I did blank screen minus intact object, would that show me the regions associated with intact object, or would it show me something else?
So, the signal that you get will be very symmetric because the arrangement is very symmetric. So, what we want to be able to pinpoint whether it's intact object, scrambled object, or blank.
To differentiate better whether it's intact object or just the visual input that we're getting, is why we have a scrap.
Very good. So we want to know, we want to make sure that the signal that we are getting from the intact object is because of the intact object only and not due to the visual input, right? So that is why we scrambled it. What is your name?
Joshua.
Joshua. Okay. All right. Okay, so now we have three different conditions, right? Blank screen, intact object, and scrambled object. So what do you think will happen between, like, if I looked in the visual region or some regions, how do you think my signal response will be? What would happen during blank screen? Like blank screen, there should be just noise. Then during visual intact object, there would be a signal increase, right? During scrambled object, what would you expect to see? Someone who hasn't answered? Yeah. Okay. 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 So what you're saying is like blank screen will be low, intact object will be very high, scrambled object will be somewhere in between. Very, very good. So that is typically what we would expect, right? What is your name? Pari? Paridhi. Okay. All right. Okay. So when we design fMRI experiment, I guess today afternoon, that is what we are going to do. So these are some of the things we have to worry about, right? So we have to put the subject, get consent form, safety screening, right? For safety screening, what do we have to worry about? Anyone? No metals, right? What happened? No, right. Yeah. And then, right, so we would be taking different scans, and then we typically first collect the anatomical images, and these are typically done with high spatial resolution. Again, what I have written is not correct. It should be maybe 1 by 1 by 1 spatial resolution, and the functional is perhaps 3 by 3 by 3, right? And so these are some of the terminology which is used. We can have axial, sagittal, or coronal slices, and then we can display them in different ways, right, with a field of view. I think my computer froze. 1 second. Okay. Yeah, so from the field of view and matrix size, we can find the spatial resolution, right? So if your field of view is the region that you're covering, and matrix size is the dimension of the matrix, and from the field of view, you can find the spatial resolution, right? So the spatial resolution is, if your field of view is 19.2 centimeter and your matrix size is 64, then to find the spatial resolution, you just divide the field of view with the matrix dimension, right? So it would be 192 millimeters divided by 64, which is 3, right? 3 by 3. Now, if my field of view was 192 millimeter and my matrix dimension was let's say 192 by 192, what would be my spatial resolution? My field of view is 192 millimeter and my matrix size was 192 by 192, right? What would be my spatial resolution? Okay. If I wanted a spatial resolution of 2 millimeters, right, what would be my matrix size? How much? Okay, how much is that? For this one? Yeah. So how much is 192 divided by 2? 96, right? Yeah. Okay. So again, like based on this is how you can design your spatial resolution and so on, and you can also change your sampling rate, right? So if you want to study for you know, if you want 200, like 136 volume and sampling rate is 2 seconds, then it is 136 times 2 seconds, right? So that's how you can design your experiment. So this is how the blank screen with intact object versus scrambled object looks like, right? I think I'm a little slow, so I'll try to cover, right? So let's say I design a task, right, where there are two conditions, like condition 1 and condition 2, and I want to know which regions there is a change, right? How would I go about doing this? Subtract what from what? To, right. So you can do that, but is the can you think of any other way? So when you do a subtraction, you're making an assumption, right? What is that assumption? That the mean value of condition 1 and the mean value of condition 2 is different. What is that test called? What is yeah, who said t-test? T-test, right? So this is called the t-test. So that is one easy way to do. And so you can have like changes for condition 1, changes for condition 2, and then you can, right, show this, right? And that the two conditions are different, and you can generate a statistical map and then say that when you have these two conditions, this brain region shows up, right? And other brain regions don't, so only these regions are associated with the task that we presented, right? So this is called the t-test, right? So this is like another way you can map the time series, right, and then display them. So while t-test is like a popular method, but one limitation of t-test is that it doesn't use the temporal information, right? So a test which is a test which can use the temporal information is called correlation, right? So you can use correlation analysis to also identify, right? How much time I have? Okay. Can I go till 12:30 and then stop? Okay. All right. So as we design the different experimental paradigm, like these are some of the variables that we use. One is independent versus dependent variable. One is categorical versus continuous. Like, you know, like some stimulus cannot be like tapping your finger at, you know, if you said 2.1 beats per second, I cannot, like most people cannot. Also for contrast, like experimental versus control, parametric versus subtractive, between versus within subjects, and then confounding factor, randomization, and counterbalancing, right? And then when we design the task, what we are trying to do is we want to control the timing and quality of the cognitive operations to influence brain activation, right? So we may have a hypothesis as to when we want to study this region using this paradigm and what would we expect. And so that is what we are trying to do. So then we have to decide what is the best way to activate that region, right? How to optimize this. In terms of what we can control, right? We can control the stimulus properties, right? Like if we were to study intact object versus scrambled object, like whether to use a green pepper versus a picture of Shah Rukh Khan, which one do you think is better? Yes. Which one do you think is better? If you wanted to study object, right? Or let's say, would you use green pepper as a stimulus, or your favorite actor or actress's face as a stimulus? Green pepper. Why green pepper? Okay, but let's say, okay, I get it. So let's say I wanted to study faces, right? Some random person's face versus a movie star's face. Which one would you study? Why?
There might be a faster or longer activity.
Okay, very good, right? So that's how you can design your experiment, right? So this is like the basic study, right? So we have to have a psychological hypothesis, then neuronal hypothesis, and then a hemodynamic hypothesis. And when we are designing experiment, a lot of times we forget like the hemodynamic hypothesis. And when we design the experiment, we want to maximize both the detection and estimation, right? So which would increase variance in signal compared to baseline, and maximal variance in stimulus testing, right? Between the two conditions, you want large variance, right? And Rahul mentioned, when we design, we should also make sure there is no refractory effects, signal saturation, or habituation, or even signal predictability, right? So if I was to design a study where you had to predict what would come and I'm just using a coin toss, then the chance that you will get it right is 50%, right? So we have to have a better design. In terms of fMRI design, there are many differences in which we can present. One is called the block design. Like everything I showed so far was block design, right? Like 20 seconds for condition 1, 20 seconds for condition 2, 20 seconds for condition 3, if there is a condition 3, or else you have like 2 blocks, right? Which you repeat. The second one is event-related design, right? And in event-related design, you have specific, very short period of like where you know exactly it would be for like 5 seconds or 2 seconds. And the second one would be maybe between 1 and 3 seconds, but that period is randomized, right? It's called jittered. And the third condition is the mixed design, which we use some of this. Again, these are like the block design and event related. There are other conditions, which is like movie watching, like continuously you are doing something. I'm going to cover movie watching much later, but now we would just cover these paradigms, right? So block design, as I mentioned, is like, you have condition A, condition B, this keeps repeating, right? And this is the most popular way to study many of the tasks. It has several advantages. Um, the simplicity in design, and because the design is simple, analyzing the data is also simple, right? You know that the first 20 images are always rest, the next 20 images are always task, the next 20 are always rest, so you can do many different kind of analysis to analyze this data, right? In this, what is typically done is you also have like a rest condition, right? You could have task one rest, and repeat it, or else, like I showed you for the scrambled object, it could be task one rest, task two rest, and keep repeating, right? So the other parameter is like how long or short of a duration it should be, right? Should it be 10 seconds? Should it be 20 seconds? Should it be 1 minute, right? Um, let's say you were to design a task for finger tapping, would you use 10 seconds, or would you use 20 seconds, or would you use 40 seconds? Which one? Or would you use for 5 minutes, right? Like, so let's say you have 4 choices, right? The first one is all is block design. Rest is the first one. Rest is 5 second on, like off. Then 5 seconds of finger tapping. The second one is rest is 10 seconds. 10 seconds of finger tapping repeating. Then is, let's say, rest is 40 seconds. Finger tapping is 40 seconds repeating. The fourth condition is 5 minutes of rest, 5 minutes of finger tapping, repeating a couple of times. If you had to design, which one would you choose? You said 20, 10? Okay. Anyone else? 40, okay. No one would choose 5 seconds of rest and 5 seconds of, why? Why? Right, good. Also, what Rahul mentioned, right? Like, if you have a subject tap for 5 seconds, they would be tired. Um, so this is how like a finger tapping paradigm looks, right? For 40 seconds, 20 seconds, like 8 seconds and 6 seconds, right? Knowing, so knowing this, if you had to choose, if you knew this is the answer, which paradigm would you choose? Right, so this is 40-second. Okay, all of you can see it, right? 40 seconds, 20 seconds, 8 seconds, and 6 seconds. Which one would you choose? But for 20 seconds, you can also see that. Hmm? Okay, actually, I mean, Anyone else?
So for this one, both 679 and 7 were fine because there will be less confounding during the shorter.
And, and anyone else? Yeah, 20 seconds. But why would you choose? So in 8 seconds, right? It also comes almost to the baseline, right? And the magnitude doesn't seem to change. And if we, Polima said, Polomy, okay, all right, I'm sorry. As Polomy said, what did you say?
The contrast mark.
Yeah, no, about the repeatability. Yeah,
Yeah. So I think in this case, because we're talking about finger tapping.
right,
So, because it's a repetitive task.
right.
So if we take the 8-second one, we will be able to capture the start and stop of all the finger tapping that we're doing for 8 seconds compared
Okay.
to the 20-second and 40-second that we're doing, and also the signal will die down. The signal will not die down like 40 seconds or 20 seconds because in those cases they may be 40-second one. There will be a saturation point, but after which the signal will drop. I think that will not happen.
Okay, all right. Okay, so in this case, perhaps like 8 seconds or 20 seconds or even 40 seconds would be okay. Actually, in this case, any, like I would say, like 6 seconds is a little difficult for two reasons. One is it doesn't come to the baseline. What is the other reason? You? What is the other reason? Someone who hasn't spoken today? Be you? Yeah. If you look, the answer is in the y-axis. So the magnitude doesn't reach the peak, right? So you're getting a response, but the response hasn't saturated, right? So that is why perhaps I would not use the 6 seconds one for simple task, right? But if the task is complex, maybe I would still use it, right? Again, I should also say that I'm not a cognitive neuroscientist, so I'm more like my background is in engineering, so I do like very simple tasks. So in this case, which one would you use and which one would you not use? So in this case, it is 15 seconds, 10 seconds, 4 seconds, and 2 seconds. Why would you use 2 seconds? Would you use 4 seconds? No, okay. All right. So again, the same reason, right? So these are again like some of these conditions for block design, right? So block design allows us to differentiate between activation during two different conditions, right? However, it does not allow identification of activity common to both tasks, right? Because you have two different conditions. And then, you know, it shows you activity associated with the task, but also with block design, you can have other effects, right? So let's say the design, instead of a finger tapping, I was giving you electric shock, right? Or a pain stimulus. And I hadn't told you that it would be a pain stimulus. Although you cannot do that task, you would get into trouble with your institution. But let's say I had an IRB and I could do that. So the first time you got the pain stimulus, you didn't know, right? But the second time, right, what would happen? You most likely would, you know, move, or your heart rate and respiration would become much higher. Also, as I think Rahul had mentioned, for some other task, it could be easier for you, and if you can make that prediction, so then the response is not going to be uniform, right? So you have to be careful how you design your stimulus, right? So that is like, these are some of the things. But one thing we also have to think about is that, like, when we are doing two conditions, what is the baseline, right? And do we have a true baseline, right? And this is just showing changes in CBF, CMRO2, which is the metabolic rate of oxygen consumption, and the oxygen extraction fraction across different slices of the brain, and showing that even between them there is a lot of heterogeneity, right? And not all brain regions have the same response, right? So this is fairly heterogeneous. So when we do a task, many of uh, the reasons how much signal change would occur during the task is dependent on these factors, and not just on the stimulus design, right? Because these factors would govern how much change we would have. Right? Also, in many experiments, activation can be greater in baseline condition than in task condition, right? Again, like, if I told you that you were to get like a pain stimulus, maybe during the baseline condition, like, you would be at a much alert state, right? When am I going to get the stimulus? Is it going to be, am I going to get hurt? Is it going to hurt a lot? And so on, right? Compared to when you get the stimulus. So these are some of the assumptions we have made, right? Like with block design, one of the assumptions we have made is that you present the stimulus, the only thing that changes is due to the stimulus itself and nothing else, right? If you say that, okay, if I'm going to tap my finger during baseline, everything is whatever is there. During task activation, all those things will be there, excepting the brain region associated with this, right? That may be true for simple task, but for more complex task, that may not be the case, right? Because depending on what the task is and what you know, for condition, a baseline condition, there may be a lot of activity, right? And uh, right? And somehow the figure didn't show up. I'll try to show later. So this is the power of the block design, right? So for different duration stimulus, you have different response function. And ideally, what you want to do is, because this response function tells us how much signal change you will get based on the like duration of the stimulus. So ideally you would like to get as much response as you can, right? And so that is where you have to decide the stimulus duration, right? And then again, this is using a hemodynamic response function and then using the stimulus showing how the response would be. So again, what we want is the changes evoked by the task to be at different parts of the frequency spectrum than non-task evoked one, right? So when you do the block design, right, like, does anyone know the, like, what is the heart rate of a person, of a human? How much? Beats per minute, right? So it's about, in terms of second, it is about 1, right? How about respiration rate? So respiration is like 10 to 15 beats per minute, right? So no. Okay. It depends on the age and so on, but typically that's the one. So think of it that, let's say, beats per minute was like 10, for example, right? And you designed a stimulus which had the same frequency as your respiration rate. What would happen to your response? It, it could be masked, that's one possibility, but what could be the other possibility? Mm, maybe, maybe not. So, something very simple. Mm, yeah. Good. So if your respiration rate is the same as your frequency, stimulus frequency, you won't be able to differentiate the respiration from the stimulus, right? So that is why, like, we have to be very careful when we design the block design. So these are some of the limitation. It's sensitive to signal drift. Poor choice of condition baseline may preclude meaningful conclusion. And many tasks cannot be conducted repeatedly, right? Because there would be habituation, so on. Also, with the block design, it is difficult to estimate the hemodynamic response function. So to minimize some of these effects, there is something called the event-related design, right? So in event-related, right, so let's think of the face example, right? So in the face example, we had unknown faces, and we could look at the changes. But let's say one of you had this hypothesis that you want to see brain regions between known face, like familiar faces versus unknown faces. Then how would you design that experiment, right? So you could do a block design, but another way would be where you could have like stimulus for very short period of time, right? So you have like maybe known faces, unknown faces, and other things, and each one would be present for very short period of time, and then you can analyze them to find very specific question, right? Um, is that clear? Yeah. Okay. So okay. Um, so some experimental tasks are naturally event related. Again, like if you want to study complexity of a specific design, like for example, if I wanted to study between simple finger tapping versus I tell you to tap in a different order, like 1, 2, 4, 3, right? And give you a different order each time, then maybe a event related would be better, right? So some experimental tasks are naturally event related, and this allows for studying of trial effect. So you can look at, let's say, between simple task and complex task, or easy versus difficult, or how does the brain activation changes with habituation. Then for those kind of things, it allows us to do trial effects, like based on trial to trial changes. So this one has better relationship to behavioral factors. In terms of analysis, one can do selective averaging, but generally general linear models are used, right? And so this is like a paradigm, 500 milliseconds, then there is 18 seconds of rest, then again 500 milliseconds, 18 seconds of rest. And you can do this. How do you think the response will be for this ones? Do you think the response will be the same as you would expect in block design? The amplitude, will it be the same, or will it be less? Will it be more? Okay, so here the duration is only 500 millisecond, right? In the previous slide, I showed you that, even when we are showing for 6 seconds, it's not reaching the peak, right? So in this case, the response will be much less, right? So in this case, the duration, first of all, the amplitude of the response is much less. That's one. Secondly, in block design, condition 1 and condition 2, each one is 50%, right? Half the time is spent in condition 1, half the time is spent in condition 2. In here, what happens? Only half a second is spent in condition 1, 18 seconds is spent in condition 2, right? So if we were to do t-test, it won't be very easy, right? We need a lot of repetition to get good detectability, right? So event related, to get the same power of detection, you need to collect much more data. Okay? And so again, this is for periodic one, right? And then I had showed, right? And then this is how it shows for ISI is the interstimulus interval, right? So this is 20 seconds on, 20 seconds off. This is 8 seconds on, 2 seconds off, right? 12 seconds on. So with complexity, you won't be able to differentiate signal changes. Another option which is used is that this 18 seconds has been randomized, right? So instead of repeating every 18 seconds, you randomize it and then analyze the data. The advantage with this one is that it is very hard for the subject to predict the stimulus, right? So you can present complex stimulus, and even if the subject knows what the stimulus is, there would not be any, it's much difficult to have habituation effect, right? Because you don't know when the onset time is, right? I think I will just stop here. If you have any questions, I'll try to answer them. Like can I like take 2 minutes to talk about resting state? Yeah, just 2 minutes, right? Okay. Right. So again, I'm going to cover this again tomorrow. But this is like, so far what we have studied is that you have like a task-based stimulus, like rest is 20 seconds, activation is 20 seconds. You repeat this and you get a signal change, right? And there are many different ways to analyze the data, like t-test or correlation, and you can identify statistical change, right? How much change there is. And then you use a threshold, right, on this one, and then say that, okay, when you do this task, this region lights up, right? So for my PhD, I wanted to look at what is going on in the brain during rest. And so this is how the signal looks like. So this is the frequency spectrum outside the brain. So it's quite random and much less. In the gray matter region, this is, you can see this frequency. Can anyone tell what frequency this is? We just talked about it. Heart rate, right? About 1 per second. You can see the harmonics. You can see the respiration signal. And there was this low frequency, right? And this is how the signal looks like if you look at the sensory motor cortex, okay? And so what I did was I analyzed the data in exactly the same way as was done using task activation. So this is using task activation. When you tap your finger, this is the one. And when you use just the signal during rest, this is what we get, right? Again, this is, the subject is not performing any task, and we can still acquire or identify the sensory motor region, right? Since all of you are student, this paper of mine got rejected 4 times from different journals, and it took me two years to publish it, right? But you know, anyway, this is my most cited paper. It has more than 12,000 citations. So because people defined functional connectivity as correlation between different brain regions, Carl Friston has defined as functional connectivity, and we were observing this during rest. We called it resting state functional connectivity. So some of, again, there are, like, task activation has many advantages, but because from one scan we can identify many different regions of the network, and so that is why I feel that this method has become more popular. Also in many cases, right, like if you wanted to study infant brain function, you cannot ask them to perform a task. Or if someone was, in a stroke, coma, Alzheimer's disease, and many other, you cannot have them perform tasks. So from one scan, you can identify all these networks, right? So in the next couple of days, I hope to, present more and get more feedback. If anyone has any questions, you can ask me. Okay, all right, thank you. Yeah. There will be a quiz in the afternoon, so if you have no questions, it's okay.
we get that here or shall we? The quiz you wanted to.
No, I was joking. No, even if it's a quiz, it will be a surprise quiz. Follow the event related.